Picking up from our last video where we talked about the oxidation states of carbon, I just have a few items to sew up and I was getting kind of long there. So let's make sure we take a look again and remind you that the oxidation of carbon means removing of high energy electrons in the form of hydride ions, and often a proton comes along, and replacing it with oxygens. And the more we do that, replacing high energy electrons with low energy electrons, that is oxidizing carbon. Now, often we see more CO bonds, but not always. Sometimes you can have oxidation, as in an alkane to alkene, that doesn't involve oxygen at all. One more important thing on this top of the page here, what we can say is that reduced molecules have lots of hydride ions, high energy electrons, therefore they contain the most energy. And as we strip off those hydride ions and put them somewhere else and leave behind CO bonds, we have less energy, or these molecules over here would be the least energy. And that's really important for biology. As we take in our food, it tends to be reduced molecules like fats and sugars and proteins. And when we strip away the hydride ions, that's the real fuel that we use for our body. We're left with CO2, and there's nothing left for us to do with that CO2 but exhale it, because there's no more energy to get out of that. Okay, so let's move to the bottom of the page here and test you a little bit on these principles. Here are three molecules that are going to be extremely important as we go through the next few weeks, and I'd like you to rank them 1 to 3. Most oxidized is going to be number 1, and most reduced is going to be number 3. And just for fun, let's see if you can figure out the names of these molecules. A couple of them we've seen before, and the last one we might have to help you out a little bit. So give it a pause and rank them from highest oxidation to lowest. Okay, so the most oxidized is going to be the one with the carboxylate. The next is the aldehyde, and lastly is the alcohol over here. So the one with the most energy, let's make a note of that this is most oxidized. And that means the one with the least oxidized carbon is going to be the most energy. Now, what are these called? Well, we have a 3-carbon molecule with 3 alcohols. You may remember that as our old friend glycerol, the ol being the alcohol that we learned about with triacylglycerols in the lipid chapter. Next, we have glyceraldehyde. That's one we learned in the sugar chapter, because this is a small aldo triose. And now we're going to use that one. And lastly, over here we have glycerate. And that is a reflection of the fact that often when we take carboxylic acid and change it into its basic form, we call that a carboxylate. So we're going to go ahead and call that glycerate. Now, these two molecules over here are going to make key structures in glycolysis, and you're going to have to know those structures. So I wanted you to see that there is some oxidation level difference that will come up when we start talking about glycolysis. Okay, let's move on to the next page, and we need to follow up with a description of what we do with the hydride ions. Let's make a note of that. Where do the hydride ions go? If we strip hydride ions, that is a pair of electrons and 1 proton, off of your food, and we oxidize the carbon in doing so, what happens to them? Well, we have molecules in our body that serve to accept hydride ions. And the first one I want to tell you about is NAD. It's right here. That's nicotinamide adenine dinucleotide. We actually have a beautiful adenine nucleotide here. And then we have another nucleotide up here. This is a ribose with a 5' phosphate on there. And up here we have niacin. So this is a vitamin. And let's make a note of that. This is niacin. It's one of the B vitamins. And the purpose of niacin is to accept hydride ions. This pair of electrons here can attack the carbon at the top of niacin, and we end up rearranging these electrons in this fashion. And what is that going to look like? It looks like this. We now have two hydrogens at the top there. The electrons have moved around, and we end up going down and putting a free pair of electrons on that nitrogen. Way back when, we learned that that makes it neutral when you have a free pair of electrons on nitrogen. And I like to use a little analogy here that we will use right through the final exam, because we're going to talk about these molecules a lot. I like to think of NAD, as drawn on the left side of the page here, as a basket. And this basket can hold something, and we need it to hold hydride ions. So when we put a pair of electrons and a proton on there, it's a full basket. And this basket holds exactly one hydride ion, no more, no less. And we call it something different, by the way. We call it NADH. But I like to call it NAD dot dot H, like with a colon there, because the proton that comes along is almost not the point. It is the least important thing. What's really important is the pair of electrons. And when we say NADH, we're not remembering that there's a pair of electrons in there. So I like to call this NAD dot dot H because it's all about those electrons. That is the energy of your food. And for now, we're just going to say that we store it in the NAD as NAD dot dot H. We'll come back later and talk about what we do with that. Let's go down, and now we're going to talk about a different basket. We have another basket called FAD. This is flavin adenine dinucleotide. And this is also going to be derived from a vitamin. We have this vitamin right here. This is an old friend of ours from lab. It's called riboflavin. It's a yellow vitamin. It's another one of the B vitamins. And what we have here is another case where we have a hydride ion that needs to find a place to be. It's going to attack this nitrogen. We're going to rearrange the electrons. This electron here, this pair, is not going to go to the nitrogen, but rather it's going to come out and pick up another proton. So FAD is a basket, and it has a slightly different purpose. It holds hydride ion, that's the same purpose, but it also holds an extra proton. It might seem like that's no big deal, and frankly, it's not a huge deal, but we do want to make a note that these are slightly different baskets, and they do hold slightly different contents. If we are going to draw what we end up with over here, we end up with a slightly different version of the riboflavin. That is the full basket, and we would call that FAD dot dot H2. Your books call this FADH2. I like to put in the dots because it's all about those electrons. So here we have something interesting. Vitamins that you take in your diet or you get from your food, those are going to be serving chemical purposes in metabolism. And these two B vitamins, their purpose is to hold hydride ions, high energy electrons that we strip off our food, until we're ready to use that fuel for other things, which will come later. That's the end of this video. We're going to talk a little bit about oxidation and reduction more on the next one, and other types of reactions. So let's finish this one up.